as people. Okay. Okay, so hello everyone. It's an absolute pleasure to be here today. My name is Jose McFarlane Figueroa, and I am an assistant professor at the Department of Biomedical Engineering at Columbia University. And so today I would like to talk to you about some of my work, our work on defining drug-induced landscapes using single-cell chemical genomics. And broadly, the way that the talk will be, or the webinar will be set up is that the first half will cover some of our works in chemical genomics that's previously published, and then the second half will dive into some newer work into how we're extending those platforms going forward. Now, broadly, our lab is interested in how cancer responds to therapy. Specifically, what characterizes the response of tumors that respond well to therapy versus those that are refractory, and how that heterogeneity relates to the molecular changes that are induced by drug exposure. So I'll give you an example. For example, how heterogeneity between patients gives rise to differences in the transcriptional response that occurs in response to treatment. So, we treat patients, there's some response that occurs. What's the difference between different patients and how that relates to the response to therapy? But importantly, in the context, especially of cancer, we have to remember that heterogeneity is really not only limited into that heterogeneity between patients, but also within an individual. So for example, we can have heterogeneous tumor populations, and we can imagine that these heterogeneous tumor populations might be found across multiple patients. They might not be the same heterogeneity, and each subgroup might respond differently. Now, there are many putative sources of this heterogeneity in response to therapy, and a lot of it has to do with how cancer cells acquire the characteristics that allows them to undergo that malignant transformation. So these can be enacted via mutation of different genes. For example, if we focus on, here I'm showing you the hallmarks of cancer, and we have all these processes that can change during malignant transformation. If we focus on one of these groups, the tumor suppressors, there's a wide variety, or a large number of different tumor suppressors that might be mutated in a given cancer. Now, you can imagine that different tumors might respond differently depending on which particular gene is mutated. Now, there's similar complexity when we think about treatment. So let's focus on one of the treatments that are here for targeting these different hallmarks of cancer, specifically EGFR inhibitors. There are many different types of EGFR inhibitors, some with different mechanisms of action, and you can imagine that many can have different off-target effects. So really, the response of a particular cell or a cancer cell to therapy is going to be a complex combination of the agent that's being used and the unique background of that particular tumor. Now, high-throughput chemical genetic screens allow us to explore this large interaction space between different genotypes and different compounds. So the standard or the classic high-throughput screen starts with cells in 96-well plates. For example, you can have every column be a different compound and every row is a dose. And then you can recover some dose-response relationship for these drugs. And the power of this type of technique is that you can do this for many different samples, say across different genetic backgrounds, and determine how those genetic backgrounds, how they're different in terms of their dose-response relationships. Now, the majority of these high-throughput screens have been limited in terms of their readout to either gross phenotypic readouts, such as viability or proliferation, or very specific molecular readouts. So one example that I have here is reporter expression. You can also imagine something like kinase or enzyme activity as being a readout of a given screen. Now, these type of simple readouts drastically limit what we can learn from our screens. So we won't learn much about mechanism of action or any off-target effects. Of course, you get what you screen for. Now, in addition, the majority of these techniques are what are called bulk measurements. And these bulk measurements, where you get one measurement over many cells, can mask heterogeneity that might be present between cells, so in a complex population, for example, in a tumor, or instances where even a seemingly homogeneous group of cells, upon exposure, gives rise to multiple cell states. So for example, maybe you have multiple discrete cell states that occur when you treat a set of tumor cells, or you might have a continuous response to treatment. So if you do use a bulk method, then essentially what you'd be doing is averaging over these different cell states. Now, single cell approaches can overcome these issues, and recent developments have allowed these to scale to a magnitude where they can be used for high throughput screening. In the lab, we use a strategy called combinatorial indexing, in which cells are uniquely labeled, giving a unique combination of barcodes. Now, here's one example for combinatorial indexing RNA-seq. So the way that this starts is you have cells, you parse them across wells in a plate, and then for every well, you use a barcoded barcoded oligo for reverse transcription. So essentially, you're adding a unique index to each one of these wells. You then pool all your cells together and distribute them in another set of wells, and now you do a second reaction. In this case, it's an index ligation with a second barcode. Now you have unique combinations of two different barcodes. You can do this one more time in an index PCR step. And really what you're doing is building in situ a tag that tells you, that allows you to infer that you have a single cell, because it'll be unlikely that more than one cell has a unique combination of those three barcodes. So the nice thing about this is that it does scale rapidly, and you can do millions of cellular transcriptomes. Now, I'll note that this was developed by June Cao and Malte Spillman in Jason Urey and Cole Tratnell's labs. They've recently also used this to derive these large-scale atlases of organismal development. So here I'm showing you a beautiful figure from two accompanying papers that dive into human fetal development at the level of gene expression and the level of chromatin accessibility. So this approach allows you to make these large-scale maps. But what we want to do is be able to parse that throughput across a large number of different conditions. So we set out to do just that, and we took advantage of a key observation in the lab. And this was the fact that nuclei have very high affinity for single-stranded DNA. So here I'm showing you some immunofluorescence images of cells that are on a plate, and they've been permeabilized and incubated with DAPI, which just binds nucleic acids generally, or DNA generally, I should say. And on this middle panel, these cells were incubated with a single-stranded oligonucleotide that is conjugated to an Alexa 647 dye. And you can see that there's a nice correspondence between a DAPI-positive nuclei and an Alexa 647 positive nuclei. Now, you don't get this type of staining if you don't permeabilize the cells. So we took advantage of this to build an assay for multiplex single-cell transcriptomics that we termed sci-Plex. So sci-Plex starts just as any other high-throughput chemical genetic screen. So we have cells across wells, and we treat them with different perturbations. Then we lyse cells in well in order to expose nuclei. And then we affix barcodes to those nuclei. So we're taking advantage of that affinity of a single-stranded oligo for a nucleus. And each one of these oligonucleotides has all of the goodies that you need in order to be captured by a single-cell RNA-seq, as well as a barcode that's going to relate back for every nuclei, for every cell, which well, and therefore which condition a cell was exposed to. Now, this then allows us to pool all of these nuclei together and perform one single-cell RNA-seq experiment. So now we can parse that large throughput across a large number of different conditions. Now, I'll note that this is conceptually similar to other what's called cell hashing or single-cell hashing approaches. Our method is different in that it doesn't use any specialized reagents, which is nice if you want to scale it up to the point where you want to do a very large high throughput screen. And it is made in such a way that it connects with a high throughput single cell RNA-seq method. However, one caveat is that it does necessitate access to the nucleus. Now, we first wanted to ensure that our approach could determine the effects of various compounds on cellular transcriptomes within one experiment. And to do this, we did a proof of concept experiment on A549 lung adenocarcinoma cells, and we exposed them to 4 small molecules that target regulators of transcription. So we know we should see a response, essentially. We chose an NF-kappa B inhibitor, a glucocorticoid receptor agonist, a p53 agonist, and an HDAC inhibitor. Now, here I'm presenting to you a UMAP embedding that has the results of this experiment. Now, for those of you who may not be familiar with a UMAP, UMAP is just a low dimensional representation of the relationship between cells. So 2 cells that are closer to each other are more similar here in UMAP space, are more similar in gene expression space. Now, here every dot is a cell, and they're colored by the treatment to which they were exposed, and we see that there are clear segregation depending on the drug that a cell was exposed to. So for example, this is a large cluster of cells that were treated with a glucocorticoid receptor agonist. This is a cluster that has to do with HDAC inhibition and so forth and so forth. Now we also clearly see dose-response relationships at the level of transcription. So here I've colored cells by the dose to which a cell was exposed, and we see that cells progress or move across this different UMAP embedding depending on the concentration of the drug that a cell received. So we are clearly able to observe these dose-response relationships. Okay, so we know that here, things are working, we can label things correctly. What we really want to do, of course, is we want to demonstrate that our approach can scale to a magnitude that is sufficient for high-throughput chemical genetic screening. So we devised this screen where we exposed 3 cancer cell lines, A549, which is that lung adenocarcinoma, K562, which is myelogenous leukemia, and MCF7, which is a ductal carcinoma cell line. And we exposed these cell lines to 188 compounds. Now, this compound library was designed in such a way that about half of all drugs target either the epigenetic or transcriptional machinery, where we have a pretty good expectation that we should be able to learn something about mechanism of action given exposure and given a single cell RNA-seq readout. The other half was devoted to compounds from many different classes. And the idea here is, okay, if we don't have a drug that directly feeds into transcription, are we still able to broadly understand something about mechanism of action with a single cell RNA-seq readout? So we treated these cells at 4 doses and 2 replicates, and we have a little over 4,600 unique conditions, that is unique wells. And in this particular experiment, we captured little under 600,000 single cell transcriptomes. And I'll remind you that this is essentially one single cell RNA-seq experiment. Now, as I told you, we wanted to explore whether our approach, our single cell approach, could reveal commonalities of the compounds with similar mechanism of action. Here I'm showing you a UMAP embedding that was made from the correlations of the gene expression profiles of cells that were exposed to different drugs. And now I realize it's kind of difficult to see the shapes per se, but there are 3 different shapes for the 3 different cell lines. What's more important is the colors that denote the class or the pathway that a drug that a cell was exposed to is targeting. So we see, for example, there's this blue cluster that's made up of compounds that are targeting the epigenetic machinery. This is mostly HDAC inhibitors. There's this light blue area, which is enriched for compounds that target cell cycle regulation. There's a very small, for example, cluster here that's made up mostly of HSP90 inhibitors. So we are able to capture commonalities in gene expression profiles for a large breadth of different compounds. Now, so transitioning over to the actual single cell data, here I'm showing you UMAP projections of the results of this very large screen for the 3 cell lines. And for each of these, vehicle exposed cells are in gray, and then cells that were treated with absolutely any compound are in red for K562, blue for A549, or yellow for MCF7. And what I hope you appreciate is that cells that were treated with a compound segregate to different areas of the UMAP compared to vehicle, and some do this in a cell type specific manner. So here I'm showing you the same UMAP embeddings, but now I've colored cells in blue that are exposed to the MEK inhibitor trametinib at doses that lead to a significant change in gene expression profiles as observed in clustering in UMAP space. So we identified that there were robust transcriptional changes in response to trametinib for these 2 cell lines, K562 and A549, but not so much for MCF7. So we did note this by the lack of that bright blue cluster. Now, this differential response can be explained at least partly by the genetic differences between the cell lines. So K562 and A549 carry mutations in upstream regulators of MEK kinase, which is the target of trametinib, whereas the driver mutation for MCF7 is in the PI3K-AKT pathway, so a parallel pro-survival pathway. So it does make a lot of sense that we see the strongest response with in response to MEK inhibition for cell lines that have overactivated MEK activity. Now, that's the type of result that, it is very interesting, but it's still something that we could understand or gain from a more classic bulk approach. So really what I want to highlight today are what are those additional insights that we can obtain by using our single cell resolution. So I want to highlight some examples in which our approach reveals heterogeneity in drug response. And I'll start with an example, an observation where we observe that cells respond heterogeneously to a 1 drug dose combination and induce multiple cell states. So here I'm presenting UMAP embeddings for the A549 lung adenocarcinoma cell line that were exposed to one of 2 microtubule stabilizing agents, apothelone A or apothelone B. Now, treatment with these agents led to large changes in the transcription of cells that we can visualize using UMAP. And we can see that they accumulate in 3 distinct places across the UMAP. Now, these accumulations are significantly different compared to control. So we wanted to understand what is leading to these differential responses that occur at virtually all doses of exposure. Now, these compounds target microtubules, which among other things, they are really critical for proper chromosome segregation. So we hypothesized that perhaps part of this differential distribution is due to cells responding differently at different stages of the cell cycle. So here I'm showing you that same UMAP embedding, but now every cell is colored by an aggregate score of the levels of genes that are associated with proliferation or proliferation index. So if a cell is more yellow, it's expressing higher levels of genes associated with proliferation and growth. So we do observe that 2 of these boxes are areas of fairly high proliferation, whereas 1 is enriched in an area of low proliferation. And if we break down that index even more, this box is associated with cells that are in the G2M phase of the cell cycle, and thus in G1S. So we do see that some of that differential response is at least associated with a cell being in different stages of the cell cycle. Now I want to give a second example where we observe a continuum of cellular response after treatment to an entire class of drugs across all 3 cell lines. And this allowed us to learn something about the mechanism of action by which these particular drugs act. So here are those same UMAP projections, but this time every cell is colored by the pathway that's targeted by the small molecule to which a cell is exposed to. So one of the most prominent features of this view are these very large and long projections of cells that are exposed to one of 17 HDAC inhibitors. Now, I'll note that K562 does have a similar projection, but because of the particular 3D UMAP orientation, it's actually going into the screen. So if you look at the shadow, there's actually 3 projections on this particular cell line. Now, the tips of those projections were enriched for cells that were exposed to high doses of an HDAC inhibitor, suggesting that they induce a dose-dependent trajectory. Now, to determine what defines a response and whether it's similar across all 3 cell lines, we use a mutual nearest neighbor approach to produce a consensus HDAC inhibitor trajectory. That is, that we want to take vehicle and HDAC inhibitor exposed cells and align them within a common space. And that's the movie that you're seeing right now. So in this joint space, vehicle exposed cells for all 3 cell lines land on the left. And as a cell responds, or cells respond to HDAC inhibitors, they move towards the right. And I'll show you one example. This is persinostat. We see that at this low dose of persinostat, most cells are at the beginning of the trajectory. And as we increase the dose, we start to push cells farther in. And I'll note that we also observe heterogeneity in terms of the rate at which cells move across this trajectory at one drug dose combination. So what do I mean by that? If we look at the yellow cells, MCF7, we observe there are some that are fairly early across the trajectory at this dose, and some have made it quite far. So we're observing differences between individual cells in terms of the rate at which they move across the trajectory. Now we can continue to push cells through this trajectory as we increase the dose. Now, next, we really wanted to understand what is the relationship between the biochemical efficacy of individual compounds and the ability of a cell to progress along this trajectory. Now, to ask this question, we defined a transcriptional concentration 50, or that is the concentration that is necessary on average for cells to move halfway across this trajectory. Now, here I'm showing you the relationship between that transcriptional concentration 50 and the biochemical efficacy of a compound. It's IC50. Now, I'll remind you that a lower value is a more potent compound. Now, we do see that there's a nice positive relationship between the biochemical efficacy and our transcriptional EC50. We do observe that there were some outliers, and actually it was interesting to see that when we went back into our compound library list, these were some of the most insoluble compounds in our library. So even though this is a particularly potent HDAC inhibitor, likely because it crashed out of solution, it didn't, cells weren't exposed to a high enough dose to push them into the trajectory very far. Now, we also conducted differential expression analysis across this trajectory, and we wanted to identify as what are the pathways that define progression across this trajectory. And we found that this is defined by changes in expression of genes that are associated with an exit from the cell cycle, which is consistent with the fact that these are very cytotoxic compounds, as well as an increase in genes that are associated with metabolism. And one particular type of metabolism that to us really pointed to a key role of HDACs within cellular homeostasis. Now, histone acetylation is more frequently associated with the regulation of gene expression, where acetylation is a mark of open, more transcriptionally active chromatin, and HDAC inhibitors fulfill that role of removing those acetyl groups and pushing you towards a more closed, inactive state. Now, a less appreciated fact is that histones also serve as reservoirs for cellular acetate. So they really can be thought of as well as the capacitors for the cell. And I encourage you, if you're interested in learning more about this topic, you can follow the literature of Siavash Kurdistani. This is a wonderful review that hits upon that notion. Now, acetyl-CoA derived from acetate, which is the product of HDAC class 1, 2, and 4 deacetylation, is critical for many metabolic processes. And really that inhibition of HDACs prevents recycling of those acetyl groups from chromatin for various catabolic and anabolic processes. So here I'm showing you a small pathway diagram of reactions that increase acetyl-CoA pools. And when we looked at our data, we observed an increase in expression of all of these genes towards the end of the trajectory, suggesting an effort to increase acetyl-CoA pools. So we thought that cells may have been undergoing an adaptive response to low levels of acetate. Now, however, if this molecular trajectory really is a result of acetyl-CoA deprivation that cells are trying to recover from, then we should be able to rescue, or that is block, progression along the trajectory by supplementing cells with acetate in the presence of an HDAC inhibitor. So we did that experiment. And here I'm showing you density plots of the results. Cells treated with the HDAC inhibitor persinostat, and this is at a dose that pushes the cells far into the trajectory. So here's the density plot for that particular exposure. And as we go up, we're increasing the amount of acetate. And we see that at the highest levels of acetate, we're able to block some of that progression towards the very end of the trajectory. And this suggests that at least a proportion of the sensitivity to HDAC inhibition is due to acetate deprivation, where HDAC inhibition pushes you towards an acetate-limited state and towards cell death. And we show that this could be rescued by addition of acetate, and even though I didn't show it to you, pyruvate as well. Now, in summary for this part of the talk, so we developed sci-Plex, an inexpensive sample multiplexing strategy that's compatible with high-throughput single-cell RNA-seq. We applied it to perform a chemical transcriptomics screen at single-cell resolution, and that single-cell resolution revealed heterogeneity in response to microtubule targeting and HDAC inhibitors. Now, I want to highlight, though, that the single-cell resolution of our approach, where it really shines, is in the ability to screen heterogeneous systems. So what I've shown you up until now is taking a quote-unquote homogeneous group of cells and looking for heterogeneity in response. But the resolution, of course, allows us to start with a heterogeneous system. For example, you could even imagine doing models of induced heterogeneity, say if you add CRISPR-Cas9. So you can then determine the response of these complex heterogeneous models to different types of agents. I've shown you chemical agents. You could also imagine doing biological agents or even cells, because we have the resolution to parse different cells apart. And so really for the end of the talk, what I want to do is highlight recent work where we combine sci-Plex with this particular example of starting with induced genetic heterogeneity. And specifically to introduce a new method for combined single-cell genetic and chemical screens. Now, in this new method that we named sci-Plex gene by environment, or sci-Plex G by E, we begin by genetically perturbing cells using CRISPR-Cas9. Specifically, we used a crop-seq system that was developed by Paul Dottlinger and Chris Bach over in Germany. There are other systems that you can use as well. We used the crop-seq method before and other studies of parsing the genetic architecture of cancer-associated processes. And here, what we want to understand is how does genetic perturbation alter response to chemical exposure. So essentially what we do is we begin by making these heterogeneous pools of cells, then we array them across plates, treat them with different compounds, followed by our sci-Plex labeling. And we did a couple of changes to method in order to get really good identification of the guide RNA that a cell received within the context of this protocol, because there's two pieces of information that we want here, and that is what is the treatment that a cell was exposed to, and what is the guide RNA that a cell received. Okay, so now that I've introduced the method, I want to switch gears a little bit to take a minute to introduce Glioblastoma multiforme, or grade 4 astrocytoma, which is really one of the main biological focuses of my new lab. Glioblastoma is unfortunately both the most common and most malignant form of primary brain cancer. It has an incidence rate of about 3 to 4 cases for every 100,000 individuals, And median survival is somewhere on the order of between 12 to 15 months, despite aggressive treatment that consists of surgical resection of the main tumor mass, followed by radiotherapy, chemotherapy, and more common now as well, tumor treating fields. Now, glioblastoma is a highly diffusive tumor type where tumor cells invade far into healthy tissue. So really that chemotherapy is very important to target that residual disease after resection. So I'm going to begin by introducing a known genetic dependency that we decided to probe that is pretty important in the context of glioblastoma. And that is the exposure of glioblastoma cells to temozolomide, which is an alkylating agent, which is the standard of care for the disease. Now, temozolomide toxicity is mediated by formation of O6-methylguanine lesions. However, if a protein called MGMT, methylguanine methyltransferase, is expressed, this protein can efficiently reverse this damage and give rise to the normal GC base pair. So you can imagine that if we need this lesion to be around in order to get toxicity from this compound, this is a way that cells can become resistant. However, MGMT is methylated and epigenetically silenced in about half of all glioblastoma, including the cells that we did our proof of concept experiment in. Now, if that lesion persists upon replication, you get incorporation of thymine opposite this guanine, creating a GT mismatch. And this mismatch is a substrate for the mismatch repair machinery. Specifically, it is recognized by the mut-S heterodimer, which is composed of MSH2 and MSH6, which then recruits mut-L, which is a processing complex, and exonuclease 1, which removes that thymine. However, towards the end of mismatch repair, polymerases will again incorporate a thymine opposite that guanine, creating that GT mismatch again. So this is a process that's called feudal cycling that gives rise to the accumulation of these single strand gaps. And if these go through replication, they'll give rise to a double strand break and cell death or cell cycle arrest. Now, I really wanted to introduce that pathway in order to set up our proof of concept experiment where we took glioblastoma cells expressing a system for CRISPR-mediated knockdown, specifically the dead Cas9 crab system. And we transduced it with a guide RNA library that was composed of controls, including non-targeting controls, guide RNAs that target MGMT, which is epigenetically silenced in the cell line, as well as a mismatch repair component that's not involved in this process. And we also included guides against the components of the mismatch repair recognition and processing complex. And of course, we did this in a pooled manner. And then we took those heterogeneous pools and exposed them to increasing levels of temozolomide. Now, here I'm showing you a heat map where every row is a genotype and every column is a dose of temozolomide to which a cell was exposed. And the color refers to the expression of genes that are associated with proliferation. So again, that proliferation index. So a tile that is more yellow or more green is something that has a higher proliferation, higher expression of genes associated with proliferation compared to something that's more dark blue. Now we observe that our controls, as expected, have a dose-dependent decrease in the expression of proliferation genes. And whereas cells that are perturbed for mismatch repair machinery that's involved in toxifying temozolomide maintain proliferation despite large amounts of drug. Now, this decrease in proliferation is associated with an increase in the expression of the p53 target CDKN1A, which encodes the CDK inhibitor P21. Now, and here we start to see that not all genotypes are created equal. So we see that our controls have really robust expression of P21. If we look at the perturbations for the mismatch repair processing complex, they do have some expression, whereas it seems mostly abolished in cells that are perturbed for the mismatch recognition complex. Now we can also take a more unbiased view of this data. So here I'm showing you another heat map where now columns are genotypes and rows are what we call gene modules. And these are groups of genes that have similar dynamics in our experiment, specifically those that are significantly different as a function of genotype. Now, we see that our controls do cluster nicely as a function of these gene modules, including the individual components of the mismatch repair complexes, which was really nice to see. And in addition, we also observe this intermediate phenotype for the processing complex when it goes back to expression of genes associated with PT3. So if we focus on module 4, which is enriched for genes that are induced in response to PT3 activation, we see that their lowest expression is in genotypes that are perturbed from mismatch repair recognition and highest in our control cells, as we expect. And there's this intermediate phenotype again for cells that are perturbed for the mismatch repair processing complex mut-L. And this is consistent with the fact that the recognition complex on its own can signal to the DNA damage response in PT3 to begin the DNA damage response for temozolomide. Now, okay, so this looks good in terms of our assay. We're able to identify genotypes well, we're able to identify treatments well, and the combination. But really, from this type of experiment, what we also want to ask is, if we're interested in scaling up to large screens, how can we quickly summarize and prioritize genetic perturbations that alter transcriptional dose response? So that is, how do we quickly recover the relationships between genotypes? Now, one way to do this is to calculate a transcriptional concentration 50, similar to what we did for HDAC inhibitor trajectory. But here, I've defined it a little bit differently. So that is the concentration that we need, or that we want to infer the amount of drug that is necessary to reach half of the transcriptional response of non-targeting controls. So in this particular experiment, we expect that cells that are perturbed for mismatch repair recognition should need a lot more drug to reach that point across this drug dose relationship. So here I'm showing you our transcriptional EC50 calculations. The bars are colored by the predicted sensitivities for genotypes that should be sensitive or resistant to treatment. We do see that our controls have about the same transcriptional EC50, which is really nice to see. And we see that as we go through these genotypes, cells become more and more resistant. And I'll note that this is consistent with some of my previous work in characterizing the contribution of mismatch repair components to the resistance of cells to temozolomide. So now, for the very last bit of this talk, I want to show you how we're going forward to take some of these approaches and our combined chemical and genetic screens to tackle a much bigger problem in the context of glioblastoma. And that is, this goes off of this observation of receptor tyrosine kinases as what at some point seemed to be a very good opportunity for the design of potent targeted therapies in the disease. And what do I mean by this? So recent large-scale efforts, such as the Cancer Genome Atlas, have really defined the genetic landscape of many, many different types of tumors, including glioblastoma. It's actually one of the first to be profiled by the TCGA. And these efforts, as well as some prior work, identified evidence for pretty much near ubiquitous overactivation of receptor tyrosine kinase signaling, with almost 90% of tumors having an activated mutation in one of a few components of this pathway. So it really seems that most glioblastoma tumors are increasing receptor tyrosine kinase signaling. For example, almost half of all patients have an activating mutation in EGFR. And I'll also tell you that within those, it's mostly one type of mutation, the EGFRB3. However, and really, paradoxical to this view, the response rates to RTK targeted therapy have been much lower than what is expected given this particular picture. There are various different hypotheses as to why that may be. One that we are focusing on is how targeting of these activated kinase components leads to an adaptive transcriptional response that is able to recuperate receptor tyrosine kinase signaling or some other pro-survival pathway. So what we decided to do is we wanted to identify the regulators of those transcriptional responses, and specifically the contribution of the entire human protein kinome to therapy-induced transcription. So in this particular screen, we took 3 glioblastoma cell lines, each expressing a system for CRISPR-mediated inactivation, again, that DCAS9 crab, and we transduced that each one of those cells with a guide RNA library targeting the entire kinome. And then we took those heterogeneous cell pools and we exposed them to one of 4 small molecules targeting different parts of this pathway at 2 different doses as well as vehicle control. So really what we want to understand here is how each kinase in the human protein kinome contributes to the dynamic response that's induced by these compounds in the context of glioblastoma. In total for the screen, we have about 14,000 unique conditions and over 1 million single-cell transcriptomes. And again, this is within the context of 1 experiment. Now, the first question we have is whether, is the kinome in and of itself dynamic in response to these treatments? So here I'm showing you the percent of cells in our experiment, specifically our control cells or non-targeting cells that express different kinases. So here I'm showing you the expression of aurora kinase A as a function of either vehicle exposure or one of the 4 treatments, and we treated cells at 2 doses. And what we observe, for example, is that for some kinases, such as here aurora kinase A, we observe that there's a downregulation that is significant for all 3 cell lines in response to trametinib. Now, we also identify multiple kinases that have cell type dependent changes in expression. So here I'm showing you a couple of other examples. Here I'm highlighting ERB4 or HER4 was an EGFR family member that is significantly upregulated in the presence of the PDGFR inhibitor nintedanib. It also does this in the U87NG cell line. It does not do this in T98G, and here I just highlighted another tyrosine kinase that is significantly upregulated in response to treatment, in this case, trametinib. So these responses that we see down here are some of the things that we're thinking of when we talk about adaptive transcriptional resistance. So there's been an upregulation of a factor that can recover signaling from what was originally targeted with the biochemical means. Now, again, our goal with this data set is to understand how every kinase in the protein kinome contributes to the response of glioblastoma to perturbation of the receptor tyrosine kinase pathway. Now, similar to our HDAC inhibitor story, we observed conservation in the response of glioblastoma cells to these kinase inhibitors. Here is an example of one group of genes that is similarly induced across all 3 cell lines in unperturbed cells, with the strongest upregulation mediated by the MEK inhibitor trametinib. And in 2 of the 3 cell lines are also upregulated by nintedanib, although not as much. Now, paradoxically, this signature was associated with increased receptor tyrosine kinase signaling, suggesting that there is this adaptation to overcome the effect of inhibition. Now, what we're really looking for in this screen and what we're happy to see is identify kinases whose genetic perturbation, whose knockdown, led to significant changes in these particular signatures. And here I'm just showing you a kinome tree with 11 or so, maybe 13 kinases that we identified as significantly perturbing this one gene expression signature. And we're doing follow-up experiments to confirm their involvement. Now, with that, I'll leave you with that little bit of a cliffhanger and just summarize to say that we presented sci-Plex G/E, a platform for combined chemical and genetic perturbation screens at single-cell resolution, as well as strategies to prioritize genotypes in the context of a single-cell genetic screen. And we arrive at some initial set of kinases that are involved in regulating the transcriptional response to receptor tyrosine kinase inhibition. I'd like to end by thanking many people. So we started our lab in January of 2021. So of course, I would really want to thank my postdoc advisors, Cole Trapnell and Jason Dury, and everyone at my old home at Genome Sciences, as well as my new home now at Columbia Engineering in the Department of Biomedical Engineering. And I'm very excited to say that we have a great group already of undergraduate students and master students who are helping to get the lab going, and as well as our research assistant, Melanie. So please, if you have any questions or would like to just talk about any of this work, feel free to reach out. And with that, I will take any questions that you may have. Thank you. And I assume that questions might pop in through the chat. So I guess I can give it a minute, and if there aren't any questions, we can wrap it up. Well, everyone, again, and it was an absolute pleasure to be here, and I'd like to thank you again for taking the time to view this webinar. Thank you.